This video will continue our discussion of evolutionary genetics. In the previous video, we ended with four definitions of a species, and here we will dive into these definitions a bit more. The first one we'll dive into is the morphological species concept, which defines organisms based on shared traits. This is a super common method, but it's not perfect. Several questions often linger in these species descriptions. For example, how many traits are enough to consider before deciding what is a species, and how much difference in these traits is enough to separate two species? There are also issues with the appearance of species and their actual genetic differences. Sometimes these two things just don't agree. For example, the poison dart frogs pictured here are all the same species based on genetics and actually their ability to interbreed. However, each island in this archipelago here has a distinct frog morphotype, which may lead one to believe they are actually different species on each island. In the biological species concept, the ability to interbreed is central. This definition is well-founded and would seem to have some clear-cut guardrails. This ability to interbreed is based on reproductive isolation, which can be either pre or post-zygotic. In this definition, the decision really seems binary. Either you can or you cannot interbreed, and produce viable offspring. But it's not perfect. Often, observing interbreeding or attempts at it are very difficult, especially in natural populations in hard to observe places, like the ocean. Additionally, some invertebrates, especially, can hybridize in ways that seem to be exceptions to this strict rule. Two additional issues are that it does not apply to asexual species, so basically all single-celled organisms, and we obviously cannot observe interbreeding between organisms that are extinct. So despite this definition seeming the most clear-cut, it still is not perfect. The last one that we will dive into is uses molecular features, which is to say differences in DNA sequences, chromosome structure, and even protein sequences in some cases. This technique would again seem straightforward as organisms either have a mutation in a specific position in a gene or they don't. And indeed, differences in phylogenetic trees can be more objective often than morphological differences in some cases. However, a single mutation is not enough to separate two species. We Homo sapiens all have many mutations relative to each other, and yet we're all the same species. In using molecular features, we have to decide on how much difference is enough to determine that some two organisms are two species. This actually opens up a whole can of worms, which we won't get into here. But briefly, it depends on things like the length of your genetic sequence that you're using, whether that genetic sequence is a neutral or adaptive one, and how much it varies within a particular group you are investigating. All of these factors lead to the decision of sort of what percentage difference is enough for you to say two things are either the same or different. It's complicated.